Okay, seems like we're back again. Okay, dear, for our next talk, we would like to introduce our first guest speaker. She is Dr. Claudia Solís-Lemus. And her talk is, what are phylogenetic networks and why should we care? Her talk is going to be 15 minutes long, followed by a Q&A session of 5 minutes. You can record or share materials about this talk. Dr. Solís-Lemus will be receiving questions in English and Spanish, and she's comfortable with answering them in both languages as well. So Dr. Solís-Lemus is an assistant professor at the Wisconsin Institute for Discovery and the Department of Plant Pathology at the University of Wisconsin-Madison. So Dr. Solís-Lemus, whenever you feel ready.
Okay, hi, thank you, Natalia. Can everybody hear me well?
Yes, we can hear you.
Okay, so I'm gonna go ahead and share my screen then. Okay. So, can you see my slides? Well?
Yes, we can.
Yep.
Okay, thank you for confirming that. Yes, so I want to start by thanking the organizers for inviting me and obviously for organizing this event. My name is Claudia. I mean, Natalia already introduced me, and I'm going to be talking today about phylogenetic networks. And I know that perhaps there are people in the audience that are not familiar with phylogenetic networks. So I'm going to start in the first part of the talk trying to answer 4 main questions. Basically, what are phylogenetic networks? Why do we need to care about them? How do we get them? And when do we need to get them? And if there's time at the end, I might need to rush a little bit to show some comparative methods on networks that has been more of a recent work. Okay, so what are phylogenetic networks? I think we're all familiar with the idea of phylogenetic trees that are this representation or evolutionary history of some organisms. A network is simply an extension from the tree where we add new edges, here depicted in green, that represent any form of gene flow or reticulation event. By that, I mean it could be hybridization, horizontal gene transfer, introgression. So biologically, there are many possibilities for this transfer of genes from one population to another. Mathematically, we are all representing them with this extra edge and this network graph. In order to give a biological interpretation, we need to have more information because mathematically they're all represented the same way. So what we would say, for example, in this case, is that the bluefish or the genetic material of the bluefish really could be traced to different origins. So some proportion of the genes got transferred through this path, the green path, and other genes, hopefully the majority of the genes, follow the standard tree path. But other than that, everything else is just like a tree. So we still have a root, we still have some time progression from past to present. The internal nodes still represent ancestral species. So it's, if we know how to interpret trees, the idea that I want you to keep is that we know how to interpret networks. And it's similarly to how we don't really expect speciation to happen instantaneously, but we still represent it like this in a tree. Similarly, these gene flow events is not like we expect that they happen just on a single time point. So this arrow could actually represent perhaps a path of migrations or a longer time scale. But again, mathematically, we're making the simplification of just depicting this event as one arrow. Okay, I want to highlight that there are in literature, or perhaps you will see in literature, 2 main classes of networks out there. The explicit kind, which is the one that I just described. There is underlying tree. We know how to interpret it biologically. And it has also something that I did not mention. It has another parameter that we denote the inheritance probability that represents the proportion of genes that get transferred through this path. The explicit network, like I said, it has an evolutionary interpretation. There are other types of networks in literature called implicit networks or split networks, that you lose some of that interpretation because the internal nodes, they no longer represent ancestral species. You cannot root them. Even if you have an outgroup, you lose the time direction of the edges. And you don't have that clear distinction between what is speciation and what is hybridization event. The reason why you might see them in literature is that because they're really fast to be reconstructed, unlike explicit networks that they need a lot more computational time and a lot more data. But I think at the end of the day, they're worth the investment because you get something that you can interpret in evolutionary terms. And I should clarify that the bulk of my work is on explicit networks, and that's when I'm going to be focusing today. Okay, now why do we need to care about them? So there might be people that say, well, actually, I don't really care about those gene flow events. All I really care is the main signal of my data, the main vertical signal of my data, which is perfectly fine. There is no problem with that. But what we showed in a paper, I was going to say recent, but I just realized it's 2016. What we showed in a paper was that if there was such gene flow event and we ignore it and we use a tree method, we might end up with the wrong tree. And it will have a very high bootstrap support. So that's the scary part. So I'm going to just show you some simulations that we did for this paper in which we simulated data on the network here on the top right corner. So it is a network because it has this reticulation event, the arrow. And the gamma probability represents the proportion of genes that we simulate that get transferred through this arrow. So we basically simulated data on this network. So we know that it comes from a network. And then we forget that they came from a network and we use a tree method. So we compare three options. We compare concatenation, Astral, and NJST. So concatenation did not perform well in these simulations, so I'm not going to discuss it. I'm going to just focus on Astral and NJST for now. They're both coalescent-based methods in which you first go from sequences to gene trees, and then from gene trees to species tree. So what we would love is for the tree method to reconstruct the main tree, that is the tree without the arrow, right? That would be our hope. And it turns out that if gamma is small, for example, gamma is 0.1, then these methods do reconstruct the true tree. That is depicted here in white. So the white bar is measuring the proportion of times that you reconstruct the correct tree. So if we see here on the top row, as we increase the number of genes or our sample size, we are converging to the true tree. So this white bar is increasing. So the majority of the time, we recover the correct tree. But if gamma is 0.3, that doesn't happen anymore. So if you see, as we increase the number of genes in our sample, this white bar really is going to 0. To the point, for example, with NJST, if you have 1,000 genes, you never reconstruct the true tree. So you will get a wrong tree that has almost 100% bootstrap support. And the scary part of this is that I cannot give you a threshold. So I cannot say, if your gamma is less than 0.2, you'll be fine. You can use a tree method. No, we don't know it. So the value of gamma where this phase transition happens, it's unknown. It depends on the topology, it depends on the data, it depends on the organisms, it depends on so many things that we really don't have an idea. So the short conclusion at this point that I want you to take is that networks are good. They allow us to interpret other biological events like reticulation or gene flow. Explicit networks are better because they have an actual evolutionary interpretation. And if there is gene flow in your data and you ignore it, you might end up with the wrong tree. Okay, so now I've convinced you all, we all want to estimate a network. How do we do it? So let's pretend that we have whole genomes for our species in our data that are represented by these gray bars. So the first step, like I said, this is a coalescent-based method. So we first need to identify what we call coalescent genes, or in simpler terms, just these chunks of genome that are orthologs and there's no recombination inside. There are many methods to do this identification. One of them is created by my PhD advisor, Cecilia Neve. And let's pretend that you already did this. So you go through your alignments, you run MDL, you get these chunks. And I should say that these genes, I call them genes or loci, but they do not have to be protein coding genes. They're just basically any region in your genome. And then you go and estimate gene trees for these regions. There are many methods to do this. In particular, I'm highlighting Bayesian approach or maximum likelihood approach that tend to be highly used. But after that, let's pretend that we did this for a variety of genes in our genome. So now we have a sample of gene trees. And you will see that these gene trees will not necessarily match. And that's okay. So there are different reasons why there could be this gene tree discordance. One is just an estimation error, because we're just estimating them. So if our data does not have enough signal or is biased in any way, we might end up with the wrong estimate. But there are also biological reasons why these gene trees could be discordant, and such as incomplete lineage sorting that I'm not going to talk about, but also gene flow. So the idea is we want to have a method that will account for all these potential sources of variability into the reconstruction of the species evolutionary history. And this history, it could be a tree. Again, gene tree variability does not imply network immediately. It could be very well explained within a tree. So we would use a tree method like Bucky or Astral or NJST. But if you suspect that there is gene flow among your organisms in any way, then you should better use a network method, because like I showed you before, if you use a tree method, you might end up with the wrong tree. Even if you only care about the underlying tree's history, you can always ignore the arrow. You can always delete it, but then you will have a robust underlying tree structure. There are many methods to reconstruct networks, but of course, I'm going to focus on the one that I created with Cecilia Net, that's called SNAC. And SNAC is a method that takes a sample of gene trees, runs maximum pseudo likelihood estimation, and returns an explicit network. SNAC is one function within a Julia package that we created that's called phylonetworks. And if you have not tried Julia, I'd really encourage you to do so. It's kind of like if you're familiar with R or Python, it's very similar. It's just faster and better. I always say that, and I always forget when I'm being recorded. Anyway, so SNAC is one function within phylonetworks, and you get this explicit network as a result. And it is more scalable than standard likelihood. That's why it's a pseudo likelihood. We try to approximate it and make it faster. And besides speed, there are different things that we like about SNAC. In particular, we take unrooted gene trees as input. Many of the network methods will require you to root the gene trees, which could be easy or hard depending on the organism. So the fact that we take unrooted gene trees allows SNAC to be robust against rooting errors. Also, we don't take any branch lengths in the gene trees. So that prevents as well some potential violations of molecular clock assumptions or rate of evolution. So we only take gene tree topologies and they are unrooted. And lastly, one of the complaints of coalescent-based methods is that they take the gene trees as perfectly known. And we know that they are not. We estimated them. We need to propagate the estimation error. And we do that in SNAC by not taking the gene trees directly, but summarizing them into concordance factors that automatically accounts for estimation error. So SNAC is not just more scalable than standard likelihood methods, it's also more robust to different types of errors. Okay, so as a statistician, I didn't say that, I'm a statistician. I'm always worried about model identifiability, which in simple terms, I'm not gonna talk about this, but is can we detect what we want from data? Does the data have signal for what I want to estimate? And in this case, we want to estimate the presence of hybridization. I'm not gonna talk about this, but I'm just gonna refer you to a poster that you should check out of Arrigo Cohen, who's a postdoc working with me. So if you are interested to know whether we can or cannot detect hybridization events, you should check out his poster. Okay, I'm just gonna give you a motivation why you wanna care about identifiability. Basically, that tells you, again, whether the data has signal for what you want to reconstruct. So I'm going to show an example, and this will make more sense if you look at it again after seeing Arrigo's poster. But we simulated data on this network here that has two types of hybridizations, which we denote a good diamond and a bad diamond. You will see why. Here, I'm plotting on the right the proportion of times that I correctly detect the hybridization. For the case of the good diamond, I need as little as 30 genes to detect it. But to detect the bad diamond, I need 3,000 genes. So knowing this, it's crucial because it tells me that different parts of the networks will be more supported. I will be more confident in their estimation depending on how many genes I have. And this is an artifact, not just of the data, but of the characteristics of the network. So we are starting to understand what is it about certain hybridization events that make it make them easier or harder to be detected. So I think this is a very fruitful area for people interested in mathematical modeling for phylogenetics. And I think we're just beginning to make these type of studies more widespread. Okay, so one example with actual organisms, which are these fish. We use SNAC to reanalyze a data set from Ray Kui's lab, where they have 24 species roughly of swordtails and platyfish. So the swordtails have this nice sword on the tail. They have roughly 1,000 genes, and we use them to reconstruct the network here. So SNAC also gives us bootstrap support. If it's not there, it's just 100%. Here in black, it's for the tree edges. So for example, up here we have 14, 13, that's very low. So it could be that we like the data, maybe we don't have enough data, or it could be that there could be another reticulation here that we didn't reconstruct. In terms of the gene flow, we have two, one in green with 100% bootstrap support, and one in orange here, that it was jumping in the bootstrap networks. So 75% of the time, the origin of the gene flow was up here, and 25% of the time was here. In all of these cases, the recipient was Nezahualcóyotl. So we seem to be confident of who is the recipient of gene flow, but we're just uncertain of who is the origin, where is the origin. Okay, how am I doing in time? Okay, I think I'm okay. So last question from the very first part. So I showed you how do we get the network, we use SNAC, and then when. So it's not that you have to go out there now and every time that you need to reconstruct a phylogeny, you want to estimate a network. No, there is something that you could do first, and that is a goodness of fit test called Ticker. It's available on GitHub. And this is basically telling you whether a species tree is a good enough fit for your data. If you reject this hypothesis, then you want to use a network method. But if you don't reject it, you're fine with a tree model. So this is a good first step to try. Okay, so very fast so that I have some time for the questions. I have the network now. What do we do usually when we have a tree? Well, we want to study trait evolution, right? And in this case, we had two traits for the fish: sword index, which relates the length of the sword, and female preference, which were some experiments whether the female preferred males with longer or shorter swords. We just want to know how this trait evolved along the branches of the network. And it turns out that it is very straightforward from what we did with trees. We can do exactly the same things that we did with trees. Estimate phylogenetic signal, ancestral reconstruction, phylogenetic regression, phylogenetic ANOVA, everything's the same. The only thing that we're really changing is this covariance matrix. Because now in the tree, there is a fully bifurcating process that creates some form of dependency. And now here we have some branches that join again to create a hybrid. So that just creates a different pattern on the dependency. So this is a different paper that we published in Systematic Biology. And just one example here, we reconstructed the ancestral state for the sword, and we found that the common ancestor likely had a sword, which is interesting in this group that has some didn't have the sword, some had. So there's always the question of whether it was there and some lost it or it was not and some gained it. So again, just every time we do something methodologically, we want to go back and test and see whether the conclusions make sense biologically. Okay, last thing, just an advertisement again. This is my package Phylo network since in Julia. SNAC is the functions to estimate the network, but there are many other functions for network, including the comparative method functions. So you can estimate trait evolution as well on networks. We have step-by-step tutorials and a Google user group, and you can always email me directly. My email is in my website here on the bottom left. Okay, so finally, acknowledgements. There's a lot of people that have been working with me with SNAC, in particular Cecilia Ne, who is my PhD advisor, and just say hi to people in my lab and conclude saying that there are positions available in my lab. If anyone's interested, I'm sorry, I'm just trying to finish in time. You can contact me in my website. And sorry for rushing at the end, but I hope we have time for some questions.
Thank you, Claudia. There is one question. I have no idea if you want to check it by yourself. I'm going to read it out loud.
Okay, let me see. How can I?
You should have the Q&A chat option.
Maybe you can read. Oh no,
Okay.
sorry. Oh no. I found it. I found it. Yes.
Okay. Anyway, I'm going to read it.
Yes, please do.
What are the benefits of using 100 genes to detect hybridization compared to using genomics methods? Can you use this method with genomic data?
Yes, you can do it. So what we mean is if you have the genomic method, yes. So the only thing is that we will break it up, the genome, we will break it up in these coalescent loci, but it's basically, yeah, we can still use it. Yeah.
Okay, this another one. Phylo networks estimates hybridization, or there is a way to know if it's incomplete lineage sorting?
Yes, so this is a good question, and I didn't have time to go into the details of this, but basically phylo networks account for both incomplete lineage sorting and hybridization. And that's what makes it challenging computationally. So the fact that we're trying to distinguish what part of the discordance is well explained by incomplete lineage sorting and only add horizontal edges for the ones that are hybridization. So phylo networks, and actually this is true for other likelihood-based methods for networks, they tend to account for both incomplete lineage sorting and hybridization so that you don't get an overly complex network.
Okay. So another one, how sensitive is NAQ to gene re-estimation error?
Yes, so SNAC is, we've done many simulations, and of course, every method is sensitive to estimation error in a way, but we found that it depends on how you get the concordance factors that I didn't have time to get into many details. They're more in the website, but basically, we have a way to estimate the concordance factors that account for estimation error already. We are not taking the gene trees as perfectly known, which would be bad because they're not. So it's let's say it's more robust than methods that use the gene trees directly, but it's still obviously not perfect. It still suffers from bias with estimation error.
Okay, good. Let's take the last two ones. One by Mark Simmons. It says, Claudia, can SNAC accept input gene trees with polytomies? Sorry. Also, it is computationally tractable on a matrix of, say, 100 taxa and 4,000 loci?
Yeah, great question. So we do, I mean, absolutely, gene trees with polytomies are perfectly fine. The number of genes is not a problem. So SNAC can scale very well from 100, 1,000, 10,000 genes. It doesn't matter the number of genes, it scales well. The challenge is the number of taxa. And I'm sad to say 100 might be pushing it for SNAC. We have only tested on as large as 50. But I should also say I'm working on methods to make it more scalable. So I'm hoping to have something by the end of the summer or early fall, another method that will try to make even SNAC faster so that we can account for bigger data sets. At the moment, we have not tested for 100 taxa.
Okay, let's take the last one. We are running out of time. Okay, I got lost.
How confident is the method in detecting gene flow at very deep divergence? So we have found, we have not done a thorough simulation study. We want to do this actually. We have found, at least on the data sets that we tried, that we are good detecting that gene flow close to the leaves or deep, but obviously I cannot guarantee that that will happen every time. I think we need to do a more thorough study, but we have seen that it is, that it has successfully estimated gene flow on deep time scale.
Okay, good. So perhaps we should move on to the next talk. And please remember that if you want to keep discussing about this, perhaps you may use the Discord server to chat with each author of each speaker in case you have further questions.
Okay.
So thank you, Claudia.
Thank you very much, Dr. Solís-Lemus. Okay. Hello, Dr. Asher.
Howdy.
Hi. Okay, so I am going to introduce you to our next guest speaker. Okay, sorry. So he's Dr. Robert Asher, and he is going to present us the top techniques to incorporate genomic data from living species into phylogeny reconstruction of fossils without direct access to molecules. So he will be giving a 15-minute talk followed by a 5-minute Q&A session. You can record and share the materials from this talk. Dr. Asher will be receiving questions in English or Spanish, and he's comfortable with answering them in both languages. So we are very honored to have Dr. Asher here today with us. He's a senior lecturer and curator at the University Museum of Zoology of the University of Cambridge. Also, Dr. Asher has worked as an evolutionary biologist at the American Museum of Natural History in New York and the Museum für Naturkunde in Berlin. So without further presentations, Dr. Asher.
Thanks so much. I'm really pleased to be here. You guys deserve a medal for putting all this together. This is really the future, frankly. Forget about COVID. We should have been doing this a long time ago. Okay, so I hope you can see my slides. I have the title here, which you've already heard.
Yeah.
Mm-hmm. Yes, we can
And
see it.
very good. And what you're looking at there is what I will refer to as a well-corroborated tree of mammals. In a way, we benefit from our own vanity, I think, compared to other groups of animals or plants, and that more attention has been paid probably to collecting data on mammals than other animal and plant groups. And so what you're looking at is really what I would want to call truth, but the philosophers give us as phylogeneticists a hard time when we say that. Anyways, it's a well-corroborated tree showing you, I think, pretty much what the evolutionary history of mammals looks like in the form of a tree of living taxa. And it's based on, well, a very large genomic data set, microRNAs, proteomes, rare genomic events I've put here, moving my cursor slowly so you all can see it. A lot of different data sources support this tree out of an astronomically huge number of alternatives. So that's with living taxa. And we know a lot about the tree of living taxa because of comparative anatomy. I don't want anyone to think that, you know, I'm trying to tell you that a knowledge of animal evolution didn't happen until we had GenBank and the internet. Obviously, we knew a lot about that for over a century, but really the details only became clear in terms of the interordinal relationships among mammals with the genomic revolution in the 90s. And so in paleontology, we don't have these data. In paleontology, to this day, or this year at least, a wonderful fossil published by our colleague David Krause just a few months ago, a Malagasy Cretaceous Gondwanathere stem mammal, basically. Here's a paper from 2019 about another microdocodon by Zhao et al. In Science. And a couple of years ago, our colleagues Matt Barron, Paul Barrett, and Dave Norman had a lovely cover of Nature. And all of these papers and many others in paleontology, if they sample living taxa, which more often than not they don't, at least it's harder to do that with the dinosaur paleontologists because it's such a divergent stem group compared to living birds. In any event, when you do get the occasional duck or chicken or mouse or rat in some of these paleontological papers, they certainly don't include any genetic data. So you kind of see where I'm going with this. We do have very rare occasions molecular data from things like woolly mammoths or upper Pleistocene endemic South American ungulates or upper Pleistocene xenarthrans, giant ground sloths, and a wonderful compelling paper by Welker and colleagues in 2015 had 2 alpha and beta collagen chains of 2 SANUs, 2 South American native ungulates, Toxodon macrokenia, and they put forth this very provocative hypothesis that almost certainly has at least a grain of truth to it, that actually this South American radiation is a sister taxon to Perissodactyls, rhino, horse, tapir. So you do get genomic data, or at least genetic data, some kind of molecular data for some fossils, but the reality is that for our geological timescale, here's 500 million years of vertebrate history, and it's only at that uppermost limit, the strip of paint on the top of the Eiffel Tower, as Mark Twain put it. It's really only this recent period of time when we can get biomolecules for fossils. Maybe one of you is optimistic that that can change sometime soon. I'm personally not, but you know, who knows? At the moment, this is what we have to work with. But just imagine, if you would, for a moment, that we had some way of taking a genomic signal that really has revolutionized a lot of what we understand about animal and plant phylogeny, particularly mammals. And what if we had a genome for Archaeopteryx? You know, what if we had genomic data for Microdocodon? You know, what we could ask ourselves, what would the equivalent be of hippo whale if looking at Mesozoic mammals, for example, or dinosaurs for that matter? So here's finally the meat of my talk. I want to tell you what that method is: incorporate genomic signal into paleontological phylogenetics using predicted ancestors. And then at the end of the talk, I'll just spend a few minutes telling you what the implications of this method are for the phylogeny of South American native ungulates, with top terns, node ungulates in particular. So the method that I'm telling you about, predicted ancestors, was used in a paper that I wrote with my colleagues Martin Smith from the University of Durham, Amy Rankin, also from Cambridge, and Bob Emery from the Smithsonian from last year. This wasn't the first to present this method using predicted ancestors. The first, which I'll get to in a minute, was by Robin Peck and his colleague Bailey from the University of Salford in the United Kingdom. But let me just tell you what this method is. So here, what we focused on in our paper last year was this phylogenetic tree of primarily rodents and lagomorphs, or gliries. And much like mammals in general, there's a very well corroborated tree for gliries. We know where basically the living taxa belong. And it amounts to a squirrel-related clade, or what I will call Sauromorpha, Myomorpha for the mouse-related, and Tinohystrica for the guinea pig-related clade. And you'll notice that on each one of these nodes here, there's a number. These don't represent some kind of support indices. They're just arbitrary numbers telling us that the common ancestor of all of gliries is equivalent to this hypothetical predicted ancestor number 125. So, and that's a very important part of paleontology that all of us paleontologists have been, working on all of our careers. It has really comprised what paleontology means. In other words, what did animals look like in the distant past? Even Edward Drinker Cope was essentially concerned with this question. We have the advantage today, of course, of a phylogenetic framework upon which to orient our hypotheses. And of course, I'm talking about a very specific node to estimate what this predicted ancestor 125 would have looked like. So here's an artist's reconstruction of something like a common ancestor of a rodent and lagomorph. In terms of the actual method that we used in this paper last year, Beck and Bailey used in 2018, here is a screenshot of a TNT data file showing you a bunch of living taxa, some fossils, and then these predicted ancestors, 103, 104, 105, 106. These all happen to be in the mouse-related clade. So this is kind of what Pablo Golubov was talking about. And I have to say, we're very primitive in this regard. We estimate these character states just using this awful default described trees, X-out internal and Paup. Following Beck and Bailey, we go back into the reconstructed data set to just manually change an illogical ancestral character state to an inapplicable. You have, it's not an automatic procedure. You have to do that. And of course, all the problems that Pablo was telling us still apply in this case. Nonetheless, I'm just going to ignore that and carry on. And I hope it's nonetheless to convince you that this is a worthwhile method. So this is basically the method to get predicted ancestors. The key of this method is to then re-include them iteratively in another set of phylogenetic analyses. And so here's a slide where I can convey to you some of the results of that exercise of taking predicted ancestors and re-including them back into an analysis using only morphological data, right? And now, just to, in case it wasn't already clear, this tree of living taxa that I'm showing you here on the left is based on only molecular data sets, things like rare genomic events and introns and coding genes. So they're really independent data sets that converge on this tree. And several of you will have noted the various polytomies here, such as the rodent root, because it's not completely resolved, but still there's more information in here than not information. Anyways, I want to draw your attention first to these orange squares, which represents iterative sets of the certain number of predicted ancestors shown to you on the x-axis. So randomized sets of, say, 15 different predicted ancestors, doing that for just from 2 to 3 to 4 to 5, et cetera, up to the maximum possible. And on the y-axis, we have a measure of tree similarity. I want to call out to my colleague Martin Smith here, who wrote this quartet package in R, which can quantify that shared quartets. Basically, that just means similarity to the well-corroborated tree. And as you see, as you add more and more predicted ancestors using only morphological data, the similarity using just the morphology gets closer and closer and closer to this well-corroborated tree, which has no morphological data in it, right? Now, I will also draw your attention to these yellow circles, which represent real fossils. We have plenty of those in our data set, and it's not quite as steep a slope. Nonetheless, you see the similar kind of result, namely that as you add more and more combinations of fossils in various sets, you get increasing similarity to the well-corroborated tree. So now I want to switch data sets away from the rodent lagomorph Cleary's data set that we discussed in 2019. And I want to use this data set from the mammal tree of life, 4,500, give or take, morphological characters from O'Leary et al. In 2013. My colleague Juan Carillo, who is now working in Paris, got his PhD from Zurich. Him and I collaborated on a paper which took a subset of the O'Leary et al. Characters, and that's also what Beck and Bailey used. When you use the morphology by itself, without the predicted ancestors, that's PA, and without fossils, you get this very traditional looking mammal tree of life with these clades that we know aren't true, right? They're not well corroborated. So Volantia includes, say, Dermopterans and bats. In fact, Dermopterans are part of Uroconticularis, bats are part of Larisiotheres. Edentates are things like aardvark and pangolin, along with the Xenarthrans, and so on. When you add the predicted ancestors, and this is basically, I'm just repeating the results from Beck and Bailey in 2018, you add the predicted ancestors and the fossils, and using only morphological data, we recover all 4 of these well-corroborated trees. So the Larisiotheres in blue, the Uroconticularis in red, Afrotheres in green, and the Xenarthrans in yellow. And that's I don't know if I'm making this clear enough or belaboring the point, but this is a very exciting result. No genomic data here, only morphology, and we are recovering the well-corroborated tree for modern mammal groups. So now I want to tell you a little bit about some of the concrete implications that this method has for understanding one particular group of fossils, which are the endemic South American ungulates. This is a very diverse group. And just in the past couple of years, a couple of real breakthroughs have happened. Welker et al., like I mentioned before, were able to extract some collagen proteins. They're not the only one. Mike Buckley had a paper along these lines. And both of these papers supported the hypothesis that at least for the upper Pleistocene taxa, namely Macrokenia and Toxodon, which they actually sample, you get this persidactyl Sanu clade. There was a Westbury et al. Paper slightly more recently, 2017, that were able to get some, I think it was a mitochondrial genome for one of these groups. This is a figure from Welker. I think Westbury et al. Had just Macrokenia. And they also got a persidactyl Sanu clade, which is a really important and frankly, maybe revolutionary is a bit hyperbolic, but it really, no paleontologist could have told you with confidence where these animals belong up until 2015. And here we have a really positive idea. But then I want to draw your attention to a quote from Westbury et al., who say that Leptopterns and all of nodoungulates form a monophyletic unit that shared more recent common ancestry with Persidactyla. I mean, that seems to be true with the 2 taxa that were sampled, at least by Welker et al. But actually, Nodoungulata alone has 50 genera. I mean, there's probably over 100 species in this group. And so that's quite a claim, isn't it? By taking a sample of 2 and saying, well, they're all monophyletic and sister to persidactyls. I'm a bit reminded of the claim from the 1990s that guinea pigs aren't rodents. Clearly this deserves a little more scrutiny. So anyways, which brings me to the application of this method using predicted ancestors, because of course, Beck and Bailey included the South American native ungulates that Juan Carillo and I also had in our paper, which still wasn't a great sample. We only had 4 or 5 or so. And in fact, our results were different. We got the Leptoptern and one of this sort of alleged condylarth grade Sanus in the Larisiotheres next to persidactyls, but our Eocene notoungulate, so Toxodon didn't exist, of course. That's a much younger taxon. Tomosexlea is a Casamioran taxa from Patagonia, and Carodnia is an older taxon, Paleocene from Itaboraí from Brazil, and they both fell within the Afrotherian clade. And when you consider the South American radiation with the primates and the rodents, or the Malagasy radiation with the tenrecs, the carnivorans, primates, rodents there, I mean, none of these groups are completely monophyletic, right? There are very few colonization events in South America and Madagascar. So the individual radiations prefer monophyly, but the insular group as a complete total is not monophyletic. So that's one of the main conclusions of this method. I also want to shout out to my colleague, Oscar Wilson, who I hope all of you will be attending the virtual SVP in October, and Oscar has a much better data set than anything that I've put together, and so he will give you shed more light on this subject in October. So this draws me then to the conclusion of my talk. I want to show you the only figure that all of you have seen many, many times from Origin of Species. This is, of course, a phylogenetic tree, and when you have a well-corroborated phylogenetic tree, this is a gold mine for paleontologists, because just using independent bodies of data from living taxa alone, you can make hypotheses, optimize characters on it, which will benefit greatly from the problems that, or the solutions of the problems that Pablo Golubov articulated in his plenary talk. Eventually, we will know what the predictions are for all of these nodes, and then we can go to the fossil record and ask the simple question, is this what we find? And basically predicted ancestors is sort of rushing to make that discovery before we have the fossils. So I think with that in mind, I will give you this concluding sentence here, which you can read for yourselves, and I will thank you very much for your attention.
Thank you very much, Dr. Asher.
Yeah, thank you. Well, we have 3 questions here. Robert, perhaps you can check them as well as I read it. You should have the Q&A chat in the inferior part.
I'm still struggling with the Zoom format, so I'm looking.
Okay, no problem. I'm going to read it.
Yeah, go ahead. Just read it.
Okay, the first one by Gomez Rocha says, if this method could be used in other locations other than paleontology, where the access to molecular data is also impossible, such as entomology, when we have many species known only from the holotype, and most species are old and dry, so DNA extraction is difficult or impossible.
Yes. So all you're doing is, anything that's essentially unknown, whether it's a question of sampling or, restrictions in field work, or the list is, the mind boggles, right? There are lots of possibilities. So yeah, absolutely. This method is amenable to non-paleontological applications.
Okay, so the following. By Alejandra Pirot, what's the procedure of establishing predicted ancestors?
Well, there are lots of possibilities. The one I told you about was a very blunt instrument, simply using, this describe trees command in PALP. TNT is very sophisticated as well. Of course, people listening to this talk are far more expert on telling you about that than I am. All you need is some algorithm to estimate ancestral characteristics, and PALP does that. It spits out just a text file, and in my primitive ways, I just go into that and manually take a predicted ancestor and paste it into a matrix. You do have to go back and make sure, at least with the limited methods that we have now, that you're not, you will get a reconstructed state for every character just using the describe trees command in PALP. So you have to go back and change that to inapplicable, if it is indeed inapplicable. That's a problem that is relevant to any issue of missing data or inapplicable.
Okay, the next one by Martin Ramirez. Could you use a topological constraint instead of adding an ancestor?
So, I'm sorry, I was saying hi to Martin, who overlapped with me some years ago in New York. But could you repeat that?
Yeah, he asks if you could use a topological constraint instead of adding an ancestor.
Yeah, I mean, so that does lend itself to representing the genomic signal. Obviously, that's a good point, and in many ways, it's a lot simpler. You know, there are techniques to do that already. I think the issue with constraints, though, is you probably agree, is that you really want some element of data interaction. You want to enable data to reveal hidden support, for example, to use a term from John Gatesy in the 1990s. And I think with a constraint that you really limit that option, whereas with predicted ancestors, you don't. I mean, you're actually predicting this animal existed 50 million years ago, you know.
Yeah.
And it's a terminal just like any other.
Okay.
Jorge, hola, hola.
Hola. Yes, Pablo.
Querría hacer una pregunta, pero verbal, porque es larga de escribir.
Okay.
Avísame cuando pueda.
Okay, let me read one further question. Francisco Almeida is asking if the coding of the ancestor is based on the molecular topology.
Well, so a topology by itself, in my opinion, is neither molecular. I think I heard you correctly. You know, a topology is a topology, and you use a certain data set like mitogenome or rare genomic event or whatever. So the coding is based on the well-corroborated tree. It doesn't have to be a well-corroborated tree that's based on, you know, DNA. It could be based on stratigraphy in the case of foraminifera, for example, or it could be based on, I don't know, the sky's the limit. But yeah, you need the well-corroborated tree for the case that I was telling you about. Absolutely is very much the product of the genomic revolution in the last couple decades.
Okay, thank you. Pablo.
Robert, hi, Pablo Globov here. Something that
Hello.
may be somehow, hi, how you doing? Connected to what you're doing. I need to explain a little bit. As you know, I have worked a lot on tree searches, and that's the context where I did this. And I call this the fish miracle. I have tried many years ago to create all of the ancestors for a given tree, even if you take a really large and difficult data set, say, you know, SILA, 500 taxa, or, you know, 2,000 taxa for RBCR.
Yeah.
Okay. If you take a Wagner tree and do branch swapping, you never, ever get even close to minimum length.
Yeah.
Right? Okay. If you now create all of the reconstructed ancestors on a most parsimonious tree and create a matrix, the way you are doing, I believe, and now you run the matrix that now, instead of 2,000 taxa, it has 4,000 taxa.
Yeah.
You run a Wagner tree plus TBR. Almost every fucking random addition sequence you do followed by TBR gives you the optimal tree, the exact same tree which you used to produce the predicted ancestors. Okay?
Yeah,
Somehow
okay.
adding the predicted ancestors from a tree is changing the landscape, the tree landscape, making it so much, you know, more easily traversed, at least by TBR. So clearly that is introducing something in the data which is biasing the result towards the same tree which you used to produce the predicted ancestors. I wonder whether that may be a problem for the kind of approach you're doing, or whether that means that the kind of approach you're doing somehow comes almost to the same thing as using constraints, like Martin was suggesting. Okay, you may want to explore that.
Yeah, of course. I mean, all I would say is with the you know, the big difference, I guess, is the taxon numbers. We've taken deliberately just the easier problems. You know, with rodents, we had, I don't know, maybe 60, 70 taxa, Galeries, I'm sorry. And, you know, a few dozen predicted ancestors, so it was a smaller sample. And you use the term bias, of course, and that's sort of in the eye of the beholder, isn't it? I mean, the whole
okay. That may be the wrong word. That's
genomic. No, but it's perfect. It's a good word, right? But it's a bit judgmental, you know. And so the whole point is to take a genomic signal from living taxa. And we're saying in the beginning, we know what this tree looks like for the living taxa. What we don't know is where the fossils go. So in a way, we do want to bias the tree. We do want to recover Afro theory because we know that's the true, you know, the true group. So a well-corroborated group. So I'm not really changing anything that you've said. I'm acknowledging the difficulty for sure, but I think it's sort of, you need to have a willingness to acknowledge that you know, I am assuming. I mean, if someone were to come up with a new data set tomorrow that says actually all of the evidence for Afrotheria is due to incomplete lineage sorting or the stuff that we heard about from Claudia, then you know, this all goes out the window. So it could be very wrong.
Yeah, and one difference with what you're doing and what I just described is that you are not using every single predicted ancestors, or you are taking all of the ones from a tree and then you add more actual terminals, right?
Well, no, I think in this, so our data set for the Gliries, and it had about 50 extant taxa, and we took all of the internal predicted ancestors for that. I mean, the other thing that you may have noticed was that we had polytomies, you know, and that's even more problematic because we're actually saying any polytomy when reconstructing a predicted ancestor is essentially a hard polytomy, and probably that's, you know, that's a big assumption, probably a wrong assumption. So that will reduce the number of internal nodes, of course, and it's simply a factor of our ignorance of the well-corroborated tree.
Yeah. Okay, thank you.
Yep, thank you.
Yeah, you did. Thank you.
Okay, well, I think we are running out of time. Remember that we have a break now of about 20 minutes. So, yep, that's it. Thank you, Robert, for your talk.
Okay, guys. Thank you very much, Dr. Robert Asher, for accompanying us today and making some space in your agenda for being with us today. And thanks also for your amazing talk. Now we are going to have a little break. This is going to be more or less a 15-minute break. And we just want to remind you two important things about the symposium, or in general about the meeting. And the first one is, please, if you're going to participate in the Discord poster session, you have to create an account and enable the feature, enable push talk in order to participate. And also, please remember the code of conduct that we have published on our website. I think we have two people who have raised their hands, so I'm gonna check if you have any questions. Can see. Do we have any more questions? It seems that we don't. Either way, guys, we will be back in 15 minutes and we will continue with the next talk. Thank you all for being here.